Alright, Assalamualaikum and very good evening to all students. Okay, hopefully all of you are fine. Okay, so today we continue our experiment. Okay, actually experiment number 3. Okay, it's about plasmid extractions. Okay, so please be ready with your manuals. So we'll start our lab session from now on. Okay, okay. What's meaning of plasmid at all? After all, okay, introduction. Okay, what is plasmid? It's a small circular extra chromosomal structure of bacteria. Okay, so we say kalau awak tengok bacteria, for example E. coli ataupun any kind of bacteria, you will found one structure, okay, circular extra structure, which is bulat, okay, small, in between our gene structure. Okay, kalau buka balik video, buka balik structure gene structure of bacteria, prokaryote for example, they are what we call another small circular extra chromosomal. Dia bukannya genetic, gene structure is more on plasmid. Okay, so the main reason we have a plasmid is what? So plasmid actually is the main function for bacteria to exist. Okay, so in other words, plasmid is a genetic engineering to amplify or produce many copy of certain genes. Okay, in bacteria, for example, is very important for bacteria to alive, okay, ataupun still alive, right? Continue. This accessory genetic element typically account for only a small fractions of bacteria genome, right, corresponding roughly to the range between 1 to 200 kilo base pair, right? Of plasmid or more than 15K base pair might be characterized as a large, whereas plasmid or less than 10 kilo base pair are considered as small. Extremely large plasmid with a solid size for far beyond 200 kilo base pair are also known. Okay, so plasmid is one of the vectors, okay, they use for process of cloning. Okay, kalau buka beli nota chapter 4, chapter 3, okay, about gene expression, okay, ataupun chapter 4, okay, cloning. So you will learn lots of process of the plasmid. Okay, so today's experiment objective is to extract plasmid DNA from bacterial cells. Okay, how you extract plasmid DNA? Okay, first of all, the material that we need is the samples. So mostly we use a overnight bacterial solution, but fresh, okay. So culture bacteria dalam broth, okay, dan biarkan selama 24 hours, mostly fresh bacteria dalam masa 24 jam kita boleh dapat extract banyak plasmid DNA dari situ. So we are going to extract plasmid from that bacterial solutions. Okay, so the things that we need for to the experiments, number 1, Eppendorf tubes, okay, micropipette tips, okay, vortex, centrifuge, and spin column. Okay, so kalau awak tak tahu macam mana bentuk spin column dan sebagainya, saya akan tunjukkan video lepas ni, right? So there are chemical that we use for the process. For number 1 is buffer P1. So this one P1, P2 and 3 ni dia ada dalam bentuk kita punya apa tu kits, okay, kits for plasmid extractions. So what is P1? It's resuspension solutions, okay. So buffer P2 more on cell lysis solutions. Buffer N3 is more on neutralization solutions, okay, neutralize, okay. So quickly we go for the pattern methods, okay. A, how we prepare the cell lysate. Okay, one you have to do is you have to pipette ataupun ambil isolate 1 ml of E. coli culture, which is you done it freshly overnight, okay, that contain a plasmids. Okay, so E. coli memang ada plasmid. Okay, so kalau buka oleh structure E. coli, awak akan dapat satu kawasan gene structure dan juga satu lagi ada kawasan plasmid. So plasmid tu kita nak extract. Grown for 24 hours at 37 degrees Celsius, okay, 200 APM into the Eppendorf tubes. Okay. So collect the cell by centrifuging in a microcentrifuge at 13,000 RPM for 5 minutes. So this represents the speed for the microcentrifuge. Okay. And then you have to discard the supernatant. Okay, so bila protocol number 2 ni, bila awak spin, alright, you will get the pellet. Okay, so cell pellet represent the substance yang akan melekat pada kita punya tube, Eppendorf tube. Okay, so kita kena buang dia punya cecair lebihan, alright, and then quickly put 250 ml of cell suspension solution, which is P1, and resuspend the cell pellet by the shock vortex. So awak kena vortex, maksudnya awak kena hancurkan awak punya pellet sedikit supaya dia tercampur dalam kita punya cell suspension solutions. Okay, and then add 250 microliter of cell lysis solution. So kita kena pecahkan kita punya cell pellet. So this cell pellet represent our E. coli bacteria. Okay, so dalam kita punya broth tu kita ada macam-macam medium, bahan-bahan tu. So cell pellet tu it represent our E. coli, right? So that E. coli kita kena lyse. So how we lyse? We added 250 microliter of cell lysis solution or P2. Okay, so mix by inverting the tube for 4 times. So do not vortex. So invert tu maknanya kita goncangkan dia, bukan goncang, dia lebih kepada kita terbalikkan. Gunakan dua jari awak tu, jari telunjuk dengan jari telunjuk dan juga ibu jari awak. So awak cuma pusingkan dia ke atas ke bawah. Okay, tak perlu goncang. Okay, awak cuma pusingkan dia. Alright. So terbalikkan gerakan jari awak tu selama 4 kali. And then we have to incubate for 1 to 5 minutes at room temperature until the solution will be cleared. Okay, so it means that bila awak buat je 250 microliters of cell suspension P1 and plus 250 microliter of P2, okay, solution awak akan jadi keruh. Alright, so if you incubate between 1 to 5 minutes, so your solution will be cleared, right? And then you have to put 350 microliter of neutralization solutions and immediately mix by inverting the tube several times. Again, awak kena invert, bukan vortex. Right, vortex tu kita lebih kepada hancurkan dia, right? Tapi untuk invert kita just gunakan jari. So mostly vortex lebih kepada penggunaan alat, ada vortexer nama kat situ, awak boleh jumpa dia, right? Dekat makmal, okay, tapi awak tak ada buat makmal, so awak boleh akses dalam internet, nanti awak boleh tengok video dia, right? And then you will get the white precipitate form. Okay, and then you have to spin at the full speed of the microcentrifuge. So full speed means 13,000 RPM. So this one you have to edit the time for 10 minutes. So this one you have to get the precipitate pellet. Okay, so assemble a spin column while you are waiting. Okay, so how you purify the plasmid DNA? Okay, awak dapat dah simple tu. So carefully discard the clear supernatant into the spin column. Okay, so spin column tu do not include any white materials. Okay, insert the column into the collection tubes and spin maximum speed for 1 minute. So maximum speed dekat sini untuk microcentrifuge lebih kepada 13,000 juga RPM for 1 minute. Okay, so now you have to discard the flow through and reinsert the column. Okay, add 750 microliter of column wash solution, or we call it PE, and spin again for 1 minute in the same maximum speed. Okay, and let's discard the flow through. Okay, apa yang jatuh daripada bahagian bawah tu awak kena buang, discard. So the first process 7 awak spin, okay, and then awak masukkan kolom ke dalam collecting tubes, and then awak discardkan dia punya cecair dalam tu, and then you have to put 750 microliter. Okay, and then last one, you have to spin again for 1 minute, same maximum speed, and remove all the liquids. Okay. So now you have to transfer the spin column to the new sterile Eppendorf tubes. Okay. So number 13, add 100 microliter of nucleases free water. Okay, nucleases is enzyme. Okay, tujuannya adalah untuk dia menghancurkan semua nukleus ataupun gene structure yang ada. So tinggal plasmid je. Okay, to the spin column. And for the long-term storage, elute the TE instead of water. So TE ni memang ada dalam kita punya kit tu. Okay, apa kit dia? Kit yang P1, P2, N3 tadi. Ada juga bahan lain seperti PE dalam tu TE, alright. So spin for 1 minute. Okay, maximum speed juga, alright. And discard the column and carefully transfer the DNA solution to the new fresh tube and label. So to keep our plasmid DNA in the right condition, ataupun kita dormantkan kita punya plasmid, it must be stored at negative 20 degree Celsius. Okay, so awak dapatlah plasmid extraction daripada bacteria cultures. Okay. Continue. So there are post lab question you have to answer. Okay, number 1, define plasmid. Alright. Number 2, plasmid naturally exists in the bacterial cells. Okay, list the advantages of having plasmid. Okay, so kenapa bakteria perlukan plasmid? Number 3, differentiate between plasmid and chromosomal DNA. Right? Okay, all right. Is there any question so far? Is there any question about today's lab session? Ada soalan tak?
Tak ada lagi.
Tak, sir.
Okay, tak ada Right. Okay, continue. Okay. So experiment number 3, continue. Digestion of the DNA. Okay. So how you digest is the process of kita punya DNA. Right. So digestion continue. Okay. So in the chapter number 4 or cloning, you will learn lots about restriction enzyme. Okay. So what is restriction enzyme all about? Okay, it's referred to restriction endonucleases. Okay, endo means that inside nucleus. Aces is our enzymes, but it's restricted. So means that this type of enzyme will cut only the size of the size of interest. Okay, so let's say we have, for example, from the question in the future kan, kita ada EcoR1 ataupun BMH1, dia akan potong size yang sama je. So that's meanings of restriction. Endo is inside, nucleases is nucleus, aces is enzyme. So enzyme that digest inside the nucleus. Okay. So enzyme that recognize short, specific, often palindrome. Okay. So what is meaning of palindromics? A DNA or sequence that reads the same in the both directions. That's meaning of palindromics in the terms of DNA sequence. Okay. The sites of many restriction enzyme that cuts DNA are palindromes. Okay. They cleave double-stranded DNA. So DS represent double-stranded. Okay. At a specific site within or adjacent to their recognition sequence. Restriction enzymes will cut into two ends, which is sticky ends and the blend end. Okay. So from this experiment, the objective, okay, number one, we have to cut the plasmid vectors, okay, isolated in experiment number 2 with the restriction enzyme. So experiment number 2 adalah kita punya eksperimen yang kita buat tadi tu, alright, extraction of plasmid, okay. And then to cut the DNA extraction isolation in experiment number 1 with the restriction enzyme. So DNA extraction yang pertama ni adalah kita punya chick cells. Plasmid vector kita dapat pada eksperimen plasmid extraction tadi, right? So now we are going to cut our plasmid vector dan juga DNA extraction. DNA, okay. So material that we need is plasmid DNA and DNA samples. So DNA sample come from our, is it bacteria, DNA, eksperimen minggu lepas ataupun kita punya chick cell, human cells, okay. Plasmid DNA, so have we done the process of isolation isolating of the plasmid DNA. So we need appendix tubes, micropipette and tips, okay. So the chemicals we're using a restriction enzymes, sterile distilled water and buffered. Okay. So method, prepare the following reaction in the sterile 1.5 ml appendix tubes. Okay, so dalam appendix tube tu awak kena letak apa So number one, you have to put 10 times RE buffer. So RE represent the restriction and enzyme. Okay, restriction enzyme buffer is about 2.0 microliter. Okay, so from here awak letak plasmid DNA ataupun awak letak DNA. Okay, so this one plasmid DNA or DNA samples, both 5 microliter. So 1.5 ml appendix tube. So for example, kalau plasmid DNA, awak just labelkan plasmid DNA. So another tube will be DNA samples. Okay. Enzyme you put 0.1 microliters. Okay. In total, top up will be 20 microliter. So dekat sini awak dapat 5, 6, 7, 7.1 So 7.1 ni awak kena make until 20 microliter. So 71 tolak 20 lebih kurang 13.9 okay, 12.9 water. Okay, so what type of water? Kita letak sterile distilled water. And then you have to incubate at 37 degrees Celsius for at least 1 hour. Okay, so kita definitekan ataupun kita nyatakan sebagai 1 unit of enzyme will cut 1 microgram of DNA per hours. Okay, so we assume okay, 1 unit enzyme with the 1 microgram. Okay, so kita assume ataupun kita budget keadaan 1 unit enzyme akan memotong 1 microgram of DNA yang kita letakkan itu ataupun plasmid DNA. So you have to gently flick the tubes. Do not vortex. This will denature denatured proteins. Flick tu maknanya awak ketuk aja. Awak punya 1.5 milliliter appendix tubes. Okay, and then spin down in the microfuge for a few seconds. So speed adalah maximum. Okay, so kalau ada microcentrifuge, awak punya spin tu bergantung kepada tahap yang terakhir maximum. And then place the tube in the bath water ataupun water bath for 60 degrees Celsius for 29 minutes. So the enzyme will become inactive. Okay. Spin down the content of reaction tubes and then you have to run the samples on the agarose gel. Do not forget to run with the uncut DNA samples. Okay, for what? To compare. Okay, awak dapat tak? Okay, so from here awak akan dapat berapakah size gene yang telah dipotong oleh kita punya restriction enzymes. Bisa awak letak for example, awak letak ekor R1, ekor R1 akan dapat size ekor R1 tu. Ataupun BAMH1, HI, awak akan dapat berapa size dia. Okay. So on the graph paper, plot the log molecular weights of each fragment of DNA ladder against the distance. Okay, molecular weights ni awak punya y-axis, distance your x-axis. So estimate the size of fragment in the samples. Okay, so again there are postulate question. In your opinion, what do you assume in the containing within each band? Okay, so if this were a fingerprinting gel, predict how many kinds of samples of DNA can you assume were placed in each separated walls. Okay. Number third, explain why there are more than one bands of DNA for each of the samples. Okay. Last one, name the chemical that causes the DNA to become fragmented. Okay. And number 5, conclude about the DNA samples in the agarose gel photograph based on the analysis of the graph plotted. So this one, kalau tengok question daripada Samsung Prime 5 ni, lebih kepada expected result. Okay. So you can find the result on the books or on your own. Right. Okay. So cuma saya nak bagi tahu dekat eksperimen untuk digestion ni. Okay. So the word enzyme ni awak boleh ubah. Katakan awak letak dua jenis restriction enzyme. Okay. Restriction enzyme awak letak apa tu, EcoR1 or BMHI. Okay, dua. So maknanya awak letak 0.1. Another one will be enzyme 0.1 juga. Okay, so awak cuma ubah top up air tu sampai 20 microliter berapa banyak awak letak. Kalau maknanya awak letak satu lagi enzyme 0.1 jadi 0.2. 7.2 tu tolak 20 microliter, itulah bilangan air distilled water awak letak. Dia bergantung kepada berapa banyak awak punya enzim. So mostly kalau awak nak buat lebih daripada satu enzim, so awak kena letak enzim tu 0.1 enzim yang pertama, 0.1 lagi enzim kedua, 0.1 enzim ketiga. Okay. So this one example is only one restriction enzyme awak letak lah tu. Kalau dua, awak tambah 0.1 lagi untuk second enzymes. Okay. Alright, is there any question about today experiment? Okay, is there any question about today experiment? Ada soalan tak?
No, sir.
Okay, hopefully awak faham. Okay, macam mana kita buat proses pertama, kita isolate our plasmid DNA. Number 2, how we cut our plasmid DNA or DNA extraction daripada eksperimen yang chick cell tersebut ataupun bakteria. Okay. Okay, ada soalan sebelum kita tengok video mengenai isolating plasmid DNA? Ada soalan tak?
No, sir.
Alright, kalau tak ada, okay, kalau tak ada saya akan buka video ni. It's about 10 minutes video about isolating plasmid DNA. So awak boleh tengok, after that kita boleh stop. Alright.
A plasmid is a circular, non-chromosomal segment of DNA that exists in some bacteria. Biologists can manipulate plasmids in a test tube and introduce these synthetic plasmids into bacteria. The plasmids geneticists usually use have a selectable marker such as an antibiotic resistance gene, an origin of replication, a multiple cloning site or polylinker that has many known restriction enzyme sites, and a gene or DNA sequence of interest that is inserted into the multiple cloning site. These artificial plasmids are used as vectors to replicate or express the particular genes or DNA sequences of interest that have been inserted into the plasmids. Plasmids are introduced into bacteria through a process called transformation. Although bacterial transformation is far from 100% efficient, antibiotic selection for cells carrying the plasmid of interest is very efficient. Since bacteria replicate plasmid DNA before each cell division, and since some plasmids are present in several copies per cell, a simple overnight culture of bacteria can yield vast numbers of plasmid DNA molecules. For this reason, bacterial cells can serve as factories for making large amounts of plasmid DNA and the DNA sequence of interest. In order to extract plasmid DNA from bacteria, biologists use a plasmid isolation kit, sometimes called a plasmid mini-prep kit. This kit consists of a variety of buffers and a column that binds to the plasmid DNA to purify it. Biologists begin with an overnight liquid culture of transformed bacteria carrying the desired plasmid. First, they separate the bacterial cells from the liquid media. Then they resuspend the cells in a resuspension buffer and lyse or break open the cells to release the plasmids with a lysis buffer. A neutralization buffer is added to precipitate cellular debris, but leave the plasmid DNA in solution. The tube is centrifuged to separate the solid precipitate, which will be discarded from the liquid supernatant. The supernatant containing the plasmid DNA is passed through a spin column containing a silica matrix. The plasmid DNA binds to the silica matrix, while the rest of the supernatant passes through the column and is discarded. The column is then washed with wash buffer to remove any remaining contaminants. Finally, an elution buffer is added to release the plasmid DNA from the silica matrix. This flow-through is caught in a fresh tube. The final solution contains isolated plasmid DNA. Each step of this protocol will now be demonstrated and explained in detail. Label the 1.5 milliliter microfuge tubes you will need. You will label 1 tube per culture. Using sterile 5 milliliter pipettes, pipette 1.5 milliliters of each culture into the appropriate labeled microfuge tube. Use a fresh pipette for each culture to avoid contamination. You will now harvest the bacterial cells from the culture by centrifugation. Place the microfuge tubes in a tabletop microcentrifuge and spin for 1 minute at full speed. Be sure to balance the centrifuge before starting it. This means that an identical tube containing an identical volume of solution should be directly across from your tube. Don't forget to secure the inner lid if your centrifuge requires one. At the end of the centrifugation, a small pellet will be at the bottom of the tube. This is made up of the bacterial cells that contain the plasmid DNA you want. The clear liquid supernatant is LB, the bacterial growth media, with an antibiotic such as ampicillin. Remove all traces of growth medium by pipetting or pouring out and discarding the supernatant. Don't worry, the pellet will not move as long as you don't touch it. In some cases, you may want to spin down an additional 1.5 milliliters of bacterial culture in the same tube to increase plasmid DNA yield. Resuspend the pelleted bacterial cells in 250 microliters of the resuspension buffer. Be sure to use proper pipetting technique when you do this. After you have added the 250 microliters of resuspension buffer, mix until no cell clumps are visible in the suspension. The resuspension buffer used in this step contains RNase, which will degrade RNA from the mini-prep in the next step when the cells are lysed. Once all of the pellet in your tube has been resuspended, carefully add 250 microliters of the lysis buffer. Mix thoroughly by inverting the tube 4 to 6 times. The solution should become viscous and slightly clear. Do not allow the lysis to continue for more than 5 minutes. Do not vortex at this point, or else the genomic DNA will be sheared into small fragments and will contaminate the plasmid DNA. If you are using a QIAGEN kit, the cell suspension will turn blue after the addition of the lysis buffer. Mix the solution until you have a uniformly colored suspension. The lysis buffer contains a mixture of the base sodium hydroxide and the detergent sodium dodecyl sulfate, or SDS. The SDS dissociates the lipid components of the cell. The cell membrane, which is made up of lipids, breaks open, or lyses. The contents of the cell, including the cell's chromosomes and plasmid DNA, spill out into the solution. The SDS and high pH of the solution then denature the chromosomal and plasmid DNA. Working quickly, add 350 microliters of neutralization buffer and mix immediately and thoroughly by inverting the tube 4 to 6 times. Do not vortex. The solution should become cloudy. If using a QIAGEN kit, mix until all traces of blue are gone and the solution is colorless. This neutralization buffer contains potassium acetate and acetic acid. The potassium acetate returns the pH to neutral, allowing the DNA strands to renature. The long single strands of chromosomal DNA get tangled with other cellular debris such as proteins and lipids to form an insoluble precipitate. Meanwhile, the smaller and still intertwined plasmid DNA strands quickly rehybridize and remain in solution. Now close the tubes and centrifuge them for 10 minutes at full speed. Remember to balance your centrifuge. While your samples are centrifuging, Label one spin column for each culture you started with. Each spin column consists of the column set inside an empty collecting tube. Label both the collecting tube and the spin column. After 10 minutes, a white pellet should appear at the bottom and side of the microfuge tube. This pellet consists of the proteins, lipids, and genomic DNA that precipitated out when you added the neutralization buffer. The plasmid DNA that you want is still in solution in the supernatant. The supernatant is what you want to keep. Add the clear supernatant to the spin column by pipetting or pouring the supernatant directly into the column. You can now discard the microfuge tube containing the pellet. There is no cap for the spin column, so place it carefully in a microcentrifuge. Remember to balance the centrifuge. Spin for 1 minute at full speed. During the spin, label clean 1.5 ml microfuge tubes to hold your final purified plasmid DNA. Be sure to include your initials and the date. Once the spin is done, remove the spin column and collecting tube from the centrifuge. Dump out the flow-through that is now in the collecting tube and place the column back in the collecting tube. The spin column contains a silica membrane that will tightly bind the plasmid DNA that is in the salty, buffered supernatant. Any remaining RNA, proteins, or other macromolecules pass through the membrane and are found in the flow-through after centrifugation. At this point, the DNA is bound to the silica matrix in the column. To remove the salts left over from the buffer, wash the spin column by adding 750 microliters of wash buffer, which contains approximately 70% ethanol. The salts are soluble in 70% ethanol, but the DNA is insoluble. Centrifuge for 1 minute. The insoluble plasmid DNA remains bound to the silica membrane, while the flow-through carries away the salt. Discard the flow-through. Residual ethanol from the wash buffer may inhibit subsequent enzymatic reactions, so it is important to dry the spin column with a second spin. Centrifuge again for 1 minute to remove any residual wash buffer. When you are done, place the spin column in the clean, appropriately labeled 1.5 milliliter microfuge tube. Your plasmid DNA is still on the spin column. Discard the collection tube with any flow-through. Add 50 microliters of elution buffer directly to the membrane in the spin column. Let the spin column stand 1 minute while the DNA becomes solubilized. Then centrifuge the column and tube for 1 minute. The microfuge tube caps will not close, so be sure that the caps are facing toward the center of the centrifuge so that they do not break off while spinning. After centrifugation, you should see about 50 microliters of clear, colorless liquid in the bottom of your microcentrifuge tube. This contains purified plasmid DNA. Discard the spin column, but keep and store your tube of purified plasmid DNA.
Okay, so that's a video about how you isolate the plasmid DNA from the fresh bacteria, right? So now we continue to see how the process of restriction in digestion ataupun RE enzymes we're doing or done the kita punya sample, okay? Continue.
Restriction digestion. more than 10% of the total reaction volume.